Hello doctors, today we will learn about the how urea will be formed. Let's discuss about the urea cycle. Introduction. Urea cycle is also called as Ornithine cycle. Urea is the end product by protein metabolism and 80 to 90% nitrogen containing substances excreted in the urine. How? Let's see. First, we will see the definition. It is the series of biochemical reaction that converts highly toxic ammonia into urea for the excretion from the body. Then the site. Tissue site is liver and intracellular mitochondrial matrix cytosol. First and second steps in the mitochondrial matrix and other remaining steps occurs in cytosol. Remember, tissue site is only liver. Starting product is ammonia, carbon dioxide and aspartate. End product is urea and fumarate. Pathway consists 5 main enzymatic steps. Remember 5 enzymatic steps. First is synthesis of carbamyl phosphate. In this reaction, CPS1 means carbamyl phosphate synthase first. This enzyme is used and N-acetyl glutamate NAG NH4 plus CO2 plus 2 ATP with using CPS1 plus NAG it gives carbamyl phosphate plus 2 ADP. CPS1 requires N-acetyl glutamate means this enzyme requires this NAG for its activity and it is irreversible step and a rate limiting reaction. First reaction is over, second is formation of citrulline. Here carbamyl phosphate plus ornithine forms citrulline with the enzyme of ornithine transcarbamylase. Hence, ornithine is regenerated and used in urea cycle. And citrulline produced is transferred to cytosol by transporter system. Then third step is synthesis of arganosuccinate. Citrulline plus aspartate plus ATP. It gives arganosuccinate with the using enzyme arganosuccinate synthase. Three steps are over. Fourth step is cleavage of arganosuccinate. First formation, then cleavage. Arganosuccinate get cleaved by the enzyme arganosuccinase and form arginine and fumarate. Fumarate liberated here and provides connecting link with the TCA cycle and glyconeogenesis. Do you remember that fumarate is a step fumarate converted into malate in the TCA cycle and gluconeogenesis also have importance for fumarate. Hence it is the form connecting link between TCA cycle and gluconeogenesis. Fifth step is formation of urea. Arginine plus water it gives urea and ornithine. Hence ornithine is formed in the fifth step. It is regenerated through which this second step. Here used enzyme is arginase. It is the final process. And ornithine regenerated enters the mitochondria by the ornithine translocase. Here three steps are in cytosol and from here to mitochondria ornithine go to mitochondria with using translocase enzyme. Ornithine may be considered as catalyst and arginase is mostly found in liver. Liver can be ultimately produces urea. Only liver can produce the urea. Then the summary of the urea cycle. Ammonia plus carbon dioxide plus aspartate plus NH3 ATP. It gives urea plus fumarate plus 2 ADP, 2 inorganic phosphate, adenine monophosphate and 2 inorganic phosphates. So energetics involved in urea cycle, that 2 ATP utilized for synthesis of carbamyl phosphate and 1 ATP converted to adenine monophosphate to produce arganosuccinate, which equals 2 ATP. Means here 4 ATPs are consumed. Here, since while synthesis of arganosuccinate, we require 2 ATPs. Hence, 4 ATPs are consumed in the regulation of the urea cycle. First reaction is catalyzed by carbamyl phosphate synthase first, CPS1, and it is the rate limiting reaction. It is allosterically activated by an acetyl glutamate NAG. It is synthesized by glutamate and acetyl CoA and degraded by the hydrolase. Rate of the urea synthesis in the liver correlated with concentration of NAG. High concentration of arginine increases N-acetyl glutamate concentration, and low concentration of arginine decreases the NAG. It is the mechanism how reaction is controlled. Hence, it is the rate limiting reaction. Protein rich meal increases the level of NAG and increase the urea synthesis. If you take protein rich meal that contains nitrogen containing like amino acids are there more, then urea should be formed more. Hence NAG level increases and urea synthesis also increases. CPS1 and glutamate dehydrogenase regulate each other in the formation of ammonia and its utilization for synthesis of carbamyl phosphate. Remaining 4 enzymes controlled by concentration of respective substrate. Next we will see about the disposal of the urea. Urea produced in the liver free diffuses and transport that go into blood, then excreted through the kidneys. It is the disposal of the urea. Small amount of urea enters in the intestine, breakdown to CO2 and ammonia by the bacteria, enzyme and urease. So NH3 lost in feces, absorbed in blood in two ways. Ammonia can be excreted in the feces or absorbed in the blood. In the renal failure, blood urea increases, so called as uremia, and diffusion of more urea into intestine that causes breakdown of the NH3 leads to ammonia increased in the blood. Next is significance of the urea cycle. Integration of urea cycle and TCA cycle. Urea cycle linked in three ways. How? This is called as bicyclic integration between two cycles. First is production of fumarate in urea cycle. In the previous steps pathway we have seen fumarate is formed and it is linked to TCA cycle. Important point of integration with TCA cycle: fumarate converted into malate and that's converted into oxaloacetic acid. Oxaloacetic acid transamine by the and form aspartate, and it's also enters in the urea cycle. Then aspartate plus citrulline forms arganosuccinate. So fumarate is more important for converted it into aspartate, and aspartate is important for this reaction and form arganosuccinate. ATP in the TCA cycle generated 10 ATP and in the urea cycle 4 ATP is used. CO2 liberated by TCA cycle in mitochondria can be utilized by the urea cycle. Here is the integration between TCA cycle and the urea cycle. Fumarate formed fumarate from malate to oxaloacetic acid, and then aspartate, and then arganosuccinate. Let's see blood urea. In the normal blood urea concentration is 10 to 40 mg per dl, and excreted in urine 15 to 30 gram per day. Blood urea estimation is used for screening test for evaluation of kidney functions. Elevation in blood urea. There are three main reasons, that pre-renal, renal and post-renal. Increase the protein breakdown. It is the pre-renal, and renal is acute glomerular nephritis, and post-renal obstruction in urinary tract. Azotemia, it is the clinical that increases blood urea or other metabolites which may not be associated with the renal disease. Here is one little bit point related to nitrogen compounds and urea cycle. Non-protein nitrogen, NPN, N containing other than protein, includes urea, creatine, uric acid, peptides and amino acids. In the healthy person, NPN concentration should be 20 to 40 mg per dl, and molecular weight of the urea is 60 mg. 2 nitrogen molecular weight is 28 gram. If blood urea concentration is 60 mg, then blood urea nitrogen means blood urea nitrogen is 28 mg. So 1 is equal to half of the NPN, and then it too goes there. NPN is equal to 2 BUN. Hence the normal range for ratio of the BUN to the serum creatinine is 10:1 to 15:1. It is the practical question. Remember these two formulas: blood urea nitrogen and non-protein nitrogen. Here is the overlook of the urea cycle. Carbamyl phosphate forms, then form the citrulline, then arganosuccinate, then arginine, and then finally forms urea and ornithine. Ornithine is regenerated, urea is formed, goes into bloodstream and excreted through the body. If you like it, share, like and subscribe.